sickle cell anemia. So it is an autosome linked recessive trait. So this is linked to the autosome. Okay, so it can be transmitted from parents to offspring when both the partners are carriers for the genes. So when it is transmitted, when both the partners are carriers, then it is transmitted. So here it is controlled by the alleles HBA and HBS. So the homozygous dominant or the normal gene is represented as HBAA, that is both normal, and in the case of carrier, it is called the sickle cell trait, that is HBA and HBS, and recessive, it is HBS and HBS, that is both, it is an affected, that is sickle cell anemia. Okay. Both are affected. Then it is a homozygous recessive disorder. So how the sickle cell anemia is caused? So it is caused by the substitution of glutamic acid by valine at the 6th position of the beta globin chain of hemoglobin. So hemoglobin has two chains, one is alpha and the beta, that is 2 alpha and 2 beta chains. So in the beta globin chains at the 6th position, so this is the normal hemoglobin. So the genes here are C, T, C, and here it is C, A, G. So when the mRNA, they have the corresponding, that is the complementary bases, right? So G, A, G, this is the sequence in the mRNA. So here it is GUG. So as a result of a change in a single nucleotide, right? So only a single nucleotide is changed here. So there occurs a change in the amino acid. So here it is glutamic acid, but here it is valine, right? So in the case of HPS, in HPA it is glutamic acid. So here the defect is caused by the substitution of glutamic acid by valine at the 6th position of the beta globin chain of hemoglobin. Okay, so as a result of it, this is due to the single base substitution. So one base is substituted by the other base, right? So due to the single base substitution at the 6th codon of the beta globin chain from GAG to GUG. So as a result of the single, that is a point mutation, it leads to a disorder, that is sickle cell anemia. Okay. So what are the main symptoms? So this mutant hemoglobin. So the hemoglobin is a mutant hemoglobin, that there's a change in the base sequence, right? So this mutant hemoglobin, it undergoes polymerization under low oxygen tension. So under low oxygen conditions, it causes the change in the shape of the RBC from biconcave to elongated sickle shell structure. So normal red blood cell will be spherical, right? So this is the normal hemoglobin, and here as a result of this mutation, the shape of the hemoglobin is changed from biconcave to sickle shape. So as a result of it, what happens? The sickle cells they polymerize together. So they group together and they block the blood flow. The sickle cell, it blocks the blood flow, which leads to a sickle cell anemia. So this is the main symptom in the case of sickle cell anemia, and it is a genetic disorder. Okay.